Hello everyone. Today we'll be solving Pearson EDEXCEL International GCSE Biology Unit 4B I1 paper 1B Jan 2022 paper. Question number 1. The diagram shows apparatus a student uses to compare inhaled and exhaled air. We can see the mouthpiece. We can see the mouthpiece is connected to a rubber tubing, and we can see the lime water. There is flask A and flask B. According to this particular mouthpiece, if there is a suction that is taking place, meaning that mouthpiece is used for suctioning the air will come from this particular location, it will bubble through the lime water, and then the air will get in from this particular location. If the particular person is exhaling, the exhaled air will come in this direction and it will get bubbled through the lime water, and then the exhaled air will then get out through this location. The student breathes into and out of the mouthpiece for 1 minute. Explain which flask exhaled air passes through. So exhaled air will be passing through the flask B, as we have already gone through it, because the mouthpiece is connected to the long tube which is in flask B. In the flask B, the tube from the mouthpiece is dipped in the lime water, which causes it to bubble through the lime water. All right. And we cannot inhale from flask B, because if we were to inhale from flask B, then lime water would get suctioned into this mouthpiece. Part 2. Explain the changes that will happen in the lime water in flask A and in flask B. So basically, the lime water in flask A will remain clear, because normal atmospheric air has only 0.04% carbon dioxide. 0.04% carbon dioxide. However, the exhaled air which is passing through the flask B would get cloudy more faster, because this air contains, the exhaled air contains 4.0% carbon dioxide. So 0.04% carbon dioxide in the inhaled air and 4% carbon dioxide in the exhaled air. Part 3. The student uses lime water to compare the composition of exhaled and inhaled air. Suggest an alternative substance that could be used. An alternative substance that can be used instead of lime water is sodium hydrogen carbonate solution. Sodium hydrogen carbonate solution, all right, can also be used. Sodium hydrogen carbonate indicator can also be used as an indicator for carbon dioxide in the inspired and expired air. Part B. Describe the role of diaphragm and the intercostal muscle in inhalation. During inhalation, the diaphragm contracts, which causes the diaphragm to become flattened and it moves down. And the external intercostal muscle also contracts, which causes the rib cage to be raised up and it moves out. This results in the increased thoracic, increased volume of chest cavity. And as a result, the pressure inside the chest cavity decreases. And this draws in air into the lungs. Question number 2. The diagram shows an insect pollinated flower called a lily. We can see the stigma, we can see the anther, we can see the petal, and we can see the stem which is holding the flower. Now the question says, which of these is the name of the structure P? So structure P is definitely anther because it holds the pollen. We can see that. Okay. Next question. Which of this is the name of the structure Q? So structure Q is the petal. So this is the anther, petal, and this is the stigma. On which structure do the pollen grains germinate? We know that pollen grains germinate in stigma. So the answer will be stigma. Part B. Describe how the structures PQS would differ in a wind pollinated flower. In a wind pollinated flower, the stigma needs to be feathery, more spread out, and it has to have more surface area. So the stigma will be feathery. It will be outside the leaf. It will be exposed to the air. And the anther, well, the anther will be exposed out into the air and it's going to be so that it can easily lose pollen. And the structure Q, which is the petal, wind pollinated flower does not need to attract any insect. All right, so it doesn't need to be attractive at all. The petals would be smaller. It would be probably green in color. It would not be colored or not be scented at all. Part C. Plants can also reproduce by asexual reproduction. Give one natural method that plants use to reproduce asexually. So basically, plants can produce runners, and then it can produce tubers, and it can reproduce using them. Part 2. Give one artificial method that a plant grower may use to reproduce a plant asexually. The plant, the grower can make cuttings. Part 3. Suggest why a plant grower may choose to reproduce a plant asexually rather than allowing the plants to reproduce sexually. Actually, the grower can choose to reproduce asexually because when a particular plant is reproduced asexually, it is basically a clone of the original plant. And if the original plant is a high productive plant, all of its clone will also have high productivity in terms of fruits or seeds to maintain desired characteristics. So the grower can choose to reproduce asexually. And sometimes, even if the plant produce seeds, the seeds might not be viable. So asexual reproduction is the only way. Question number 3. The diagram shows a cycle found in ecosystems. Give the name of the cycle. So we can see the cycle mentions carbon dioxide. The carbon dioxide is taken up by the plant. Carbon dioxide is produced by the plant. Carbon dioxide is produced by combustion of factory fossil fuels. Carbon dioxide is produced in respiration. All right. So all of these processes produces carbon dioxide. So this is carbon cycle. Part B1. Which process is represented by the letter W? So let's see. The letter W looks like it is combustion of fossil fuel. So it will be combustion. X is respiration. Y is also respiration. So we'll just label it like this. Okay. Z is Z represents dead or decaying. All right. And then X represents respiration. Okay. Y is photosynthesis because carbon dioxide is turning into part of the plant. So which process is represented by the letter W? So the letter W represents combustion. Next question. Which process is represented by the letter X? X represents respiration. Part C. Name a group of organisms that is responsible for decomposition. We know that fungi and bacteria, these are the group of organism. So we'll write either fungi or bacteria. Part D. Carbon dioxide is released into the atmosphere by the decomposition of organic material. The rate of this decomposition depends on a number of factors. Design an investigation to find out if changing the pH of organic material affects the rate of decomposition. Include experimental details in your answer and write in full sentences. So guys, the question said that carbon dioxide is released into the atmosphere by decomposition of organic material. So we will focus on CO2 is released. So since CO2 is released and respiration took place, obviously the mass of the material that we're trying to decompose will eventually decrease, provided that we have the same humidity at the beginning of the experiment. At the end of the experiment, loss of moisture should not be accounted for, because it's gonna be the same for the beginning of the experiment and also for the end of the experiment. Next, the rate of this decomposition depends on a number of factors. Okay, design an investigation to find out if changing the pH of organic materials affect the rate of decomposition. Since our question says that we will have to design an investigation where we'll have to find out the changing of pH of organic material, whether will that affect the rate of decomposition or not. So for that reason, we'll have to design an investigation to find out and the change of pH in the organic material. So we need organic material of different pHs. First requirement. So different pH of organic material is prepared by adding buffers so that the pH is maintained throughout the experiment. Because throughout the experiment we don't want our pH to change so that it doesn't affect our experiment. So we want a buffer to be used throughout the experiment. Same organic material and volume of organic material was used in all experiment. To keep the experiment fair, we must keep it same for all experiment. At least 3 repeats were performed at each pH so that we can calculate an average. The loss in mass of organic material is measured after each week. All experiments were performed at the same temperature, same humidity, and using similar bacteria to keep the experiment fair. Question number 4. The data in the table was collected in Japan during a 7-year study. Scientists collected data on the age of mothers and whether they smoked during pregnancy. They also recorded the percentage of babies that had a low birth mass. Now we can see age of mother in terms of year: 19 and under, 20 to 24, 25 to 29. We have different age groups. And then data for mother who did not smoke during pregnancy: number of mothers, percentage of babies with low birth weight. All right, we can see since the answer is expressed in percentage, so it's easy to compare. All right, data for mothers who did smoke during pregnancy: number of mothers we can see, and percentage of babies with low birth mass. So we can see low birth mass higher in the first column. All right, it's higher. We can see also higher for the second one, third one, fourth one. Okay, we can see that in cases, all right, it is 1.5 times higher. All right, at least 1.5 times higher or even twice as higher than the percentage of babies with low birth mass for people those who didn't smoke. Now the question says, part A, calculate the percentage. Calculate the percentage of mother age 19 years and under who smoked during pregnancy. So we can see 19-year and under, we have 1331 and 356. So we will have 356 at the top and we'll divide it by the whole number. So 1331 plus 356. And then we're gonna do a percentage into 100%. So this gives us 21.1%. So 21.1. All right. So 21.1%. Now the question says determine the ratio of non-smoker to smoker used in the study. Give the ratio as the nearest whole number n in the form of n is to 1. So in the whole study, okay, we have a number of mothers, non-smoker to smokers. So let's write down non-smoker on this left hand side plus smokers. So there's one ratio. So we do the ratio like this. We'll have to count out this entire group of number and we'll have to count out this entire group of number. So counting out all the non-smoker mothers, it becomes 85,147, and the number of mothers those who smoked total is 7,206. If we put them in ratio, in ratio with 7206, our answer comes 11.82 is to 1. Since the question says that n must be a whole number, so we'll write it down 12 is to 1. So the ratio for non-smoker to smoker is 12 is to 1. A student determines that this data concludes that smoking is the main factor that causes low birth mass. Use the data and your own biological knowledge to comment on this conclusion. We can see from this particular data that if the mother smoked, the mother had a higher percentage of babies with low birth mass in every cases. So there is no question that yes, smoking is important. The higher the percentage of low birth mass, all right, occurs due to smoking. So, guys, the question was a student examines this data and concludes that smoking is the main factor that causes low birth mass. Use the data on your own and your own biological knowledge to comment on this conclusion. Guys, if we look at this particular data, we can see the number of non-smoker mother is higher, far higher than that of the mothers who smoke. However, non-smoker mothers, the percentage of babies with low birth mass in every age group is lower than with smoker mothers, where the percentage of babies with low birth mass is higher. However, in particular, we can see if mothers were pregnant before 19 years or under, then they had a higher percentage, and if mothers, those who were getting pregnant above 35 or above, was having higher percentage of babies with low birth mass. So yes, smoking can be a main factor since all the age group smokers have low mass babies. All right, more low mass babies. There is a positive correlation with smoking and low birth mass. We can say that. However, since this particular question says that this is, we have to, comment on this particular conclusion. So we must write against it. So next, no, the smoking might not be the main factor since age of the mother affects the birth mass as well. There are more with low birth mass under 19 and above 35. There is a greater effect of smoking at older ages above 35. That can be because the mother has smoked for a long period of time. All right, because a woman who is at age 35 may have already smoked for like 15 years, or sometimes if 20 years. So the data does not give information on the mother's body mass and weight, which could be one contributing factor for the child's body mass and body weight. The table also has no information on the number of years the mother has smoked. The table provides no information on mother's health care, medical conditions, or if the mother had some kind of alcohol consumptions, or there was like diet problem, there were like drugs that were involved. So only smoking cannot be concluded. So this makes the investigation inconclusive. Further study needs to be done before concluding. Question number 5. Scientists carry out an experiment to see if reducing the availability of oxygen affects the production of yogurt. They use increasing acidity as a measure of yogurt production. They record the acidity of two cultures, one of with a reduced oxygen level and one with a normal oxygen level over 210 minutes. The table shows the result. In a reduced oxygen level, all right, we can see, all right, we can see all right 0.7. We can see there is an increase in percentage acidity from 0.2 to 0.7 within 210 minutes. With normal oxygen level, it goes from 0.2 to 0.7. So both were the same. However, we can see that with a reduced oxygen level, it reached at 180 minutes at 0.7. All right, and with normal oxygen level, it took 210 minutes. All right, now the question is, explain why increasing acidity can be used as a measure of yogurt production. We know that when yogurt is produced, then lactic acid is produced. All right, so anaerobic respiration by bacteria produces lactic acid. So that's why we can use it as a measure of yogurt production. Part B. Give one abiotic variable that the scientists should control in their experiment. There can be other points as well, apart from temperature, volume of milk, type of milk. Lactose content of the milk can also be monitored. All right, it has to be similar in all experiment. The starting pH, all right, should be similar in all experiment. The source of the milk should be similar in all experiment as well. Okay, C1. Plot a line graph to show how the percentage acidity changes over a period of 210 minutes for reduced oxygen level and for normal oxygen level. Use a ruler to join the points with straight lines. Okay, whenever we are given a graph, all right, like this, we must understand in our x-axis we are going to write, our independent variable that we can change. All right, so in this case the independent variable is the time. We can choose the time when we are going to measure the acidity level, the percentage acidity. So we will name it time. Now we're gonna see the minutes. Okay, this is minutes, so time slash minutes. Okay, and then on the y-axis we must choose the other unit that represents the dependent variable, which is the result. So dependent variable, the acidity, the percentage acidity. Okay, so for time slash minute, 0 to 210. All right, so now if we need to see how many boxes are here. So 1, 2, 3, 4, 5, 6, 7. Now since we have 210 minutes, we can make each small square having 3 units. So this can be 30, 60, and on the percentage acidity we can see it goes from 0.2 to 0.7. 1, 2, 3, 4, 5. You can see a total of 5 big ones. So we can number it, this one as 0.2, 0.4. We just divided it into 5 portions: 0.6, 0.8, and finally 1.0. Okay, now we're going to plot the graph. We'll have to do 2 graphs. One is the reduced oxygen level, the other is normal oxygen level. So we will start with the reduced oxygen level. 0, 0.2. So at 0, 0, the value is 0.2. 30, 0.22. Question number 2. Explain why the changes in percentage acidity are different in the reduced oxygen level and in the normal oxygen level. Question number 2. Explain why the changes in the percentage acidity are different in the reduced oxygen level and in the normal oxygen level cultures. So reduced oxygen level means more anaerobic respiration of of the bacteria, all right, and thus producing more lactic acid faster, so the acidity increases faster. So guys, that's all for today's video. In our part 2 video, we're gonna solve from question number 6 to 10. Thank you. Bye bye.